I request Anusha Madam, please introduce our today's speaker, Dr. Dhivya Shanmugarajan Madam.
Good morning, everyone. It is my pleasure to introduce our esteemed guest for today's session of the National Level 1 Week e-FD on emerging research trends in pharmaceutical sciences. Dr. Dhivya Shanmugarajan, ma'am. Ma'am has a highly accomplished computational biologist and currently serves as the manager scientific applications Biovia at Altem Technologies Private Limited. Dr. Dhivya Shanmugarajan, ma'am, brings with her over 15 years of rich experience across life sciences, pharmaceutical research, academia, and scientific consulting. Ma'am has developed deep expertise in molecular modeling and simulation, machine learning, cheminformatics, and multiomics data analysis, and has played a key role in R&D and pharmaceutical innovation through digital transformation. Ma'am is well versed in industry leading platforms such as Biovia Discovery Studio, Pipeline Pilot, and Material Studio, and has delivered impactful scientific solutions to various institutions and companies across the country. She has published over 50 research papers, delivered more than 75 invited talks, and contributed to numerous training programs, workshops, and technical initiatives. Dr. Divya Shanmugarajan ma'am holds a PhD in biotechnology with her research focused on network pharmacology, gene expression studies, and computer-aided drug design. Ma'am is also a gold medalist in MTech Biotechnology and holds international certifications in data science and laboratory informatics. Today, Dr. Divya ma'am will be delivering a session on a highly relevant and timely topic in drug development, scientific of toxicity prediction and drug safety using AI and ML. As the pharmaceutical industry increasingly focuses on reducing drug attrition and ensuring patient safety, early toxicity prediction has become more crucial than ever. The integration of artificial intelligence and machine learning is a playing transformative role, predictive modeling, risk assessment, and data-driven decision-making in pharmaceutical research. We are truly privileged to have her with us today to share her insights and expertise. I am very sure that her session will greatly enhance our understanding of how advanced computational technologies are shaping the future of drug safety and toxicity prediction. So without further delay, please join me in warmly welcoming Dr. Dhivya Shanmugarajan.
Yeah, thank you so much, Dr. Anusha ma'am, for a brief introduction about me. So
thank you, ma'am. It's my pleasure.
Yeah, thank you so much. And very good morning to everyone. It gives me immense pleasure to join with you all in the morning and seeing lots of participants here. So very good morning to everybody. Hope you can see me. Great. And before that, I saw the sir was giving a very good presentation and information insights about recent drug designing and discovery. I think we are moving forward to the next era of what has to be done in the drug designing and development rather than following the conventional methods. So because the world is moving fast and we need solutions in a very shorter span of time and more efficient things. This is what people are expecting from us, and industry is also looking for the same. In the same perspectives, I believe that today we are moving more on the AI ML that is really relevant, and people has to learn step by step of this process. Hope my presentation will give them insight about how to take it forward, this AI and ML, and beginning of that, and how to predict the model, and how to design the model, and how to test the model. So this is what is going to be abstract of my session, and I will try to use a demonstration. I also will share you all one of the sample paper where you can give a try with the open source tools and softwares that will give you more knowledge and then insights about, without stopping here, that will give you the more applications to you all. With this introduction and synopsis of this, I will just share my screen, and then in the next 30 to 40 minutes, I'm going to hold on the theoretical session, and followed by, we will have a demonstration session, and followed by Q&A session. So hope this is fine for everyone. And if you have any queries, feel free to text me in the chat box, and further we can connect in person also. Thank you, everyone. Let me start the session. Dr. Sham, is it good to move?
Yes, madam. Please go ahead.
Yeah, thank you so much, Dr. Sham. Thank you. Hope my screen is visible in the presentation mode.
Yes, madam. It is visible.
Yeah, thank you. Thank you so much for confirmation once again, Dr. Shyam.
Yes, ma'am.
It's okay.
Yes, ma'am.
Very significant thing, because designing the drug and understanding about the drug properties is one of the most important and significant role. On the other hand, understanding the toxicity profiling and what will happen to the system, and if we take that specific drug or in a molecule, or if you design the molecule, what is the effect of that molecule. So all these things are really important when we try to design a drug. But for according to the current scenario, so we have a very shorter span of time to make this work cost effective manner. How we do this process? We cannot go to the lab and then test within animal models. But however, when we try to do, we have to follow the OECD principles. So all these things in ethical committee, everything will take a quite more time rather than what we expect to do and perform our process, actually speaking. So how we can do this process in a much easier way, and instead of working on more than 1K of molecules, 2K of molecules, and sorting out based on their properties and understanding its efficacy, and prioritize a molecule based on its efficiency, and taking the top 10 molecules is still a challenging task, if you ask me, especially when you go for drug designing and development. Among the 10, we are not sure about it, which is having a toxicity and which is really going to work on the gender specific models. So all these things will be a quite challenging thing when we have to predict a drug molecule and understand the toxicity and drug safety, because designing a drug will be a one phase of your work, and understanding a property of the work is going to be a rest of the 90% phase of your works, actually. So plays a significant role rather than you are designing a molecules. And drug safety, again, plays a major role. The molecules or the components or the chemical molecules that we are trying to prescribe, or when you are defining for specific receptors, it should have a safety. That means whenever you take the drug, it should not cause any side effects. Taking for one disease should not emerge for the other disease. So these things need to be rightly considered when you design the drugs. That's why the today topic is about significance of toxicity prediction and drug safety using an AI ML concepts. That means understanding those concepts by utilizing a mathematical modeling techniques and executing that in a proper way, and understanding its property is going to save your time. And also the person who is going to do this process will have more insight knowledge about their molecule. Yes, because of this toxicity is arising, because of this non-toxicity is arising. So today it's going to be a session like this, how you can handle the drug safety and understand the toxicity prediction, especially when in a phase of a drug designing and development process using an AI ML techniques. Let's join with me and to know more about it. So I will introduce my company. Altem Technologies is a leading technique in a 3D printing technology, and we are into I'm mostly based on the life sciences department, where I try to handle the Biovia portfolio. Biovia, it's nothing but accessories in the past days. So Biovia portfolio will handle the different kinds of tools, including an unified lab management solutions. Now we call as ELN, and into the material research we call as a material studio. Into the drug designing platform we call as a discovery studio. Into an AI ML platforms or an imaging techniques, and developing the models or executing the new protocol we call as a tool called pipeline pilot. So these all are the major portfolio I'm handling in the national level. So national support for pan India, I'm handling the specific Biovia tools across India. So with the short introductions, I will get into that major topic, and you can see the Altem awards for over the period of years. We are a 13 years old company. So getting into drug toxicity. So toxicity is one of the major limiting factor wherever you work on it, and concentrations. These things are really important to understand the drug toxicity, actually. So toxicological study, if the person is working, say for instance, if you have a group of people working on drug designing and development field, there is always a department where the people works on toxicity screening, because they only will let you know what is the effect of, one person will let you know the what is effective concentration, the other person will let you know about at which concentration you have to stop this particular molecule. So only this concentration, only 10 mg per ml per day is allowed for a person to take it. If you exit it, it will try to create some toxicity during excretion process. So all these things are really important to understand, especially in a therapeutic potential or treating the patients. So that's the reason. So screening of toxical drugs, especially for therapeutical potential or understanding existing molecule therapeutical potential along the toxicity screening, all these things are really important when it comes for the drug safety and analysis. So how do you do this process? Say, for example, everything needs a problem statement. Say, imagine that I am just in this session, I'm giving a problem statement to everyone, and I want you all to think about it. Now I have 1 lakh molecules in my hand. So I have 1 lakh molecules in my hand, and I have given a period of only take for example, 2 hours. In the time of 2 hours, 1 lakh of molecules, I have to predict and let the person to know, or the project manager to know, what are the effective molecules that I can take it for the next level of stage. So I have only the limited period, but I have the large data of molecules, or I don't say this is large data, the quantum good amount of data, 1 lakh of molecules. I have to understand that molecule and I have to execute the process. So what are the things will help me to drive this process in a much easier way? So this is a task. So you all now know about it. So this is a task. 2 hours, 1 lakh molecules. I have to understand the drug safety process, or I want to drive to come to the conclusion that what are the molecules, or which molecules is good for treating a specific receptors, or which molecules are good to more. In general, we keep it like this. What are which molecules are good for for the next level of the process? It can be for any receptors. So if you have like this, first you have to understand the whole nature of the system. So I have 1 lakh of molecules. What I'm going to do with that? So which profile? Can I go for experimental? No, that is not possible. So can I drive it in a conventional techniques of screening 1-2 molecules in a where it is predicting? No, that is not the way. Then how do you know about that molecules, and how do you understand that molecules? That is where we are playing the role of fingerprints. What is actually fingerprints? So let me, you people are so expertise into which I'm just trying to add a value to your systems, actually speaking. So what is a fingerprint? When you draw your structures, so maybe a CH and OH, first thing you have to understand more than the 2D structure formats, the 1D structure format of the molecules are really important to understand. So I will tell that C double bond of O like this. One sec, let me get into this. Say, for instance, if I draw one chemical structure, bear with my things, I'm just trying to explain to you all to understand the concept of fingerprint, actually speaking. So if it's simply like this is a 2D structure. Say, for instance, if I write it in 1D format, I will write it in a 6 C's. Say, for example, 6 different C's, here 1 C, here 1 C, here 1 C, here 1 C. So C1, C2 to C6. For instance, I'm introducing some double bond of O here. Okay, so if I'm introducing one double bond of O or a hydroxyl group, so what it will try to show me, again, the fingerprint, that means the 1D format of this particular structure will be changing. In which position? 1, 2, 3. So in C3 position, I will write C, the bracket, I will write OH. Kindly bear with me, I am trying to explain to you all. So I will write OH, then I will write one C, then I will write one C, then I'll write one C. This completes my whole 1D format. So this is where we have to understand this concepts called fingerprints, actually. So not all whole structure is going to contribute for a toxicity. How we do for activity, say, for example, some positions. So this is a functional position R1, R2, R3. So this, because of this functional group, it is increasing activity. That's what we will study in the QSR. Similar thing, because of this functional group, it is toxicity. It is causing a toxicity. So to understand that is called toxicological fingerprint. So each and every molecule have their own toxicological fingerprint. Understanding this fingerprint is a major task, and knowing this will help you to predict or build your AI models. Actually, this is where the exact AI is behind it, actually speaking. So to know because 1D formats are very easy to do it, very easy to predict it, and the feed of your data inputs. If I have lakhs and lakhs of molecules, it doesn't going to consume the more space. That is most important AI ML models actually. When you do for a machine learning, the space constraints are really plays a major role. So profiling of certain fingerprint of a sample. This is what I was trying to explain here. Toxicological fingerprint. So because of this functional group plays in a C3 position or in a meta position or ortho position. So that makes this molecule to get a toxicity. If I remove that molecule or remove that specific moiety, then that might reduce your toxicity profiling. This is what is called toxicological fingerprint of sample. So if you know this, then what are the things which I'm going to explain in the further will be much easier for you all. That is very important. How you say for a specific moiety is inducing a biological activity. On the other hand, there is a specific moiety which is going to contribute for the toxicity. So to understand this one in a 1D format, it's called fingerprint actually. This is what I was trying to explain here. So drug as well as toxins commonly affect your nervous systems. So whatever drugs you are taking, so the toxins you are taking, the systems which is going to affect first is called nervous systems actually. So that is whatever drugs you select and take it, you have to check with all the receptors or possible receptors which is playing in the metabolic pathway and ensuring that there is no side effects for that. That is the reason we will try to work on the understanding this potential effect is really important in the drug safety analysis. So certainly this will cause a spectrum of disorders. So when you try to treat one disease, that's what I was told in the first thing itself. When you try to resolve or when you try to treat one disease, it should not lead to cause you another disease actually. It's the most important perspective in a drug safety analysis. So understand this one, it's a really important drug designing and development. So potentially understand that process or withdraw of the causal agent is most important thing in the drug toxicity analysis. So as you keep working on, now I throw you all questions. As I keep working on in the slides, you will come to know the solutions actually. Anyhow, we'll be seeing a demonstration also. You come to know what are the solutions, which is the most easiest way of achieving that. That also you come to know about it. So why testing toxicity is significant? Especially when you identify the potential adverse effects caused by any compounds or chemicals or ligands, we will try to evaluate the safety actually. So at which concentration it is affected, why it is showing a more effect in the specific systems. So these all are the things we will be trying to understand for a viability of any drug candidates. To know the process, first we will try to understand in the various systems actually. So how it is affecting the liver, how it is affecting the kidney, how it is affecting the gut. When you take the oral absorption of this specific molecule, what is the effect of that? If I take aerosol drugs, how it is absorption, where will happen? If I take drugs in a different form of intramuscular or whatever the different routes of administration, so what is the effect of that? So that is where really the pharmaceutical research is playing the significant role based on the toxicity profiling. Are they involving the different critical pathways? Is the risk assessment is including? Are they introducing a potential immunogenicity or allergenicity? So these all are things has to be considered when you design the drug or testing the specific systems actually. And even though you try to work on it, eventually what will happen, there is a more chances or less chances that could have an effect based on the gender, age, ethnic, and geographical regions. So 99.99% the drug is good enough to move on for the safety level, but at some cost, some ethnic or geographical or gender based or age based, the specific drug's efficiency can be reduced or can be increased, depends on the things. So all these things need to be considered. That's why it is not an easy task. So drug designing is not an easy task, and understanding toxicity profiling is not an easy task. But I let you know that when you try to introduce this kind of mathematical modeling, certainly it makes your works much easier and give you more insight actually. Instead of going and finding your pin in the haystacks, it gives you the much knowledge about where it is present, how it is present, because of this, what is happening. All these informations you will try to understand when you do this kind of mathematical modeling techniques. So that's a reason you have to one have to understand the drug characteristics and environment more important, because you can see in the market, it is not a latest data, it is an old data from 1992 to 20. There are 94% drugs all cleared in a phase 1, phase 2, phase 3, phase 4, IND, all filed drugs and then come to the market, but there is a side effects and toxicity, and 6% is lack of efficacy. So many drugs are removed from the market, especially based on the toxicity is leading the major position compared to other specific parameters or other physiochemical parameters. So to understand early perspectives of having your knowledge of this particular toxicity and the drug safety, will try to avoid or remove the drugs from the market actually. This is what we have to understand a lot. So different things. So first of all, we will have to define based on a high throughput screening and identifying the non-selective inhibitors, or find the therapeutic targets of different products, or repurposing. So if you take all these things into consideration, an average cost of minimum, I was trying to mention that minimal 8.4 million will be spent on each and every drug. When you take a one drug out of the market, or when you try to rework on it, it will definitely is going to consume a lots of money. So that's where we are looking for an alternative. I should not spend more money, but I have to come out with the most efficient drug, which is a no toxicity or no side effects. So these are the most important thing that has to be considered. In a smaller scale, yes, I think a smaller scale is also not acceptable. When you come for an market like a pharmaceutical companies in a very bigger market, so these things plays a major role. So each and every minute or every second of the time should be converted into in terms of productivity to understand this concept. That's where the world is actually running on at present. So just think about it. When we take most of the drugs out of the market due to toxicity and side effects, where we are actually lying and where we have to improve ourselves. So that's where the concept of AI is playing a major role. Artificial intelligence is playing a major role. By how we can achieve this, so we are going to introduce a various machine learning algorithms. So you don't have to worry about it. The machine learning is buzzwords and AI is buzzwords, but the solutions are already within us, but we are lack to see that perspectives actually speaking. So machine learning. So you don't have to worry about this machine learning. These machine learning algorithms in one on other side or other way, we studied during our school days actually speaking. These are all statistical algorithms. So for example, during our, we have a statistical papers in our UG, PG, as well as you come across during your school days also. So that means the potential application was not known to us, but henceforth try to understand this. The machine learning algorithms are not new to us. It has been spelled in a different way in your school days and as well as in the college days, in the statistics and biostatistics actually speaking. Now they have categorized into the different techniques actually speaking, the machine learning into 3 different spaces. One we call supervised. I will let you know what is supervised and how to perform the supervised also. The other one is an unsupervised learning technique. Other one is a reinforcements. So mostly So I hope you remember the problem statement. So I have only 2 hours of time. I have 1 lakhs of molecules. I have to understand the toxicity profiling. This is what my task is about. So I am driving you all in such a way how to achieve this process. How to achieve this? I told is a mathematical modeling. How to do mathematical modeling? With the help of machine learning algorithm, I am going to proceed it. What is a machine learning? So there are certain biostatistical algorithms or a technique where I can do that. What are different techniques? These are the 3 different techniques. So one is a supervised, one is a unsupervised, and also reinforcement. So what is under supervised? What are when I have a certain problem statement, which problem statement I have to take it for supervised, which problem statement I have taken for unsupervised, or which problem statement I have to take it for the reinforcement. This is where the actual task is about in a machine learning algorithm. So to know that process, first you have to understand the nature of the algorithm. Once you know the nature of the algorithm or the machine learning model, then accordingly you can take your respective inputs and you can test your inputs and you can come out with the solutions. So this is where we are going to deal about it for the next 20 minutes or 30 minutes. Now let us get into what to supervise, where we have to introduce supervise, or in which space we have to introduce supervise. And if that case is not working, when I have to introduce unsupervised. So these are things we will discuss in a few slides. We discuss about it. Then we directly get into the pattern of the testing and we understand about the process. Then we go for the demonstration. So what is ML techniques and what are the different types of things? So machine learning algorithms may involve fitting data to the statistical models. So I told you already that based on statistical models. So all the algorithms may be heuristics in nature, and understanding the traditional model of fitting, it's a goal actually. The goal of ML is to make accurate predictions. So bringing out accurate prediction is a task for each and every users. So let's see here, what are supervised? I told supervised, we have a different techniques are available in supervised. For example, regression. So quite common example, I will mention that. If you have XY data, with XY data, what you will try to fit the data to a specific models, which is best. And you try to understand the straight line process. By that, you'll come to know about what is R square value, whether it is increasing or decreasing. So when you do this one, we call this a linear regression data. And something we try to introduce a logistical regression data. When you do have more than one specific parameters and you understand it's a polynomial, more than 1 or 2 parameters, we call it polynomial regressions. So same thing, this comes under regression data. Classification. So when I do want to divide or classify the things, then there comes a role of a classification algorithms. So very simple, it's a decision trees. So how this decision trees are going to help us, what decisions I am going to take it out of it, whether it is good or bad or mutant or non-mutant. So all these things you'll try to understand from a classification algorithm. So basically, if you ask me, it helps you to categorize new observations into the predefined classes or groups or patterns that will tell you about what class category it is following, whether A category or B category or C category. You are trying to make it out. That will comes under the specific of classifications. And other is ensemble methods. So ensemble methods will be based on the how you drive it. Example, random forest models actually. So random forest model is one of the best example where you can understand a lot of code under a single forest model or a multiple model. So all these things will comes under the ensemble models. So if you say, for example, to put in a very simple word, it's a kind of a machine learning combines a multi-individual models to create a stronger, more accurate predictive models, actually speaking. So that's where this ensemble models will be present. So we'll try to use a various, including machine learning algorithms to understand this process. Say, for example, XGBoost is one of the most common algorithm will be used in the ensemble models. Similarly, Neural networks. So again, you are driving little bit inside to it. So understanding about the molecules based on neural network, especially generative adversarial networks or an RNN method or a convolutional neural network method. So deep learning of this, all these things will come to the deep learning. So this is for a supervised learning. Similarly, unsupervised learning. So we are, say for example, unsupervised learning, dimensional reduction. If I have a more number of parameters, so drug could have more than 2 lakhs parameters minimum. So it could be from an experimental techniques or from a physiological parameters or a fingerprint, whatever is it, the drug, single drug could have to end up within a more than 2 lakhs of parameters actually speaking. So I'm not going to work on all the, maybe I could take 2K parameters. So all the 2K parameters are not required for me for the analysis. So what I will do for the respective molecules, I have to reduce my data or reduce my parameters. That's where the process of dimensional reductions will play a role. So here you place in an unsupervised learning. And clustering. So clustering and clustering algorithms, like for example, k-means squares, all these things will let me know about. So under which cluster I have to place these molecules, whether following the same category or a different category. So all these things, clusterized based and supervised learning will let you know about at single point what are similar like systems are present. So aims to, this will help your people to discover the inherent patterns as well as the structures of the data within a specific category or classes. So this is where the clustering is playing a major role. So supervise, you know about it, and which one to select and how to select, train it and execute it. Say for example, when I want to reduce my parameters or to improve my quality of the model, I will try to use unsupervised dimensional reductions like a PCA, principal component analysis, and clustering to understand the inherent pattern of the structures. So by mentioning itself, now you people will get little idea about when to use supervised and when to use unsupervised. Let me show you with the help of a simple layman example, will drive you much easier. Say for example, this is a supervised learning technique. So you come across a very famous technique called SAR, structure, activity, relationship. When I give a structure, what is the effect of that activity, because of which specific molecule the activity is driving, and you're trying to understand the relationship. This is in a simple SAR example. When I introduce quantitative, that means quantitatively the structure, how it is influencing how the activity or an toxicity is profiling, so that we try to understand to the technique called QSAR. QSAR cannot drive it as like that. It wants certain algorithm to understand it. That algorithms are called supervised learning techniques. So these are the one of the categories I could mention that. Say for example, I am giving input data. So this input data might be, I got it, you can take it to two different ways. One, if you're concentrating on toxicity profiling, my input data is only based on the toxicity molecules. Now, I will apply any algorithm. For example, let's take a simple PLS, partial least square technique. Okay, it's one of the regression technique actually, PLS technique. Now I apply the algorithm, and you are having a different parameters for that. So you A log P, acceptor, donor, electros, topological descriptors, NMR data, docking data, all are considered as data actually. Even for single molecules, a single difference in atom is for two different systems, two different molecule is considered as a data when we come for data analytics, especially machine learning algorithms. So when I introduce this one, now I apply the algorithm of a more number of molecules. Say for example, when people training you in a in a single perspective of various techniques. Say for I'm letting you know what, do this one, do that one, understand this process, understand the physics. Now what you will try to know, you will train yourself in all the perspectives. That means anything, if I give a task to the person, that person has ability to do the task in a much easier way rather than the other person who is not trained. This is what is about the training set. So what will training set will do? When I input my data, the more you give the precise inputs, This is very important of data mining. So when you try to work on this machine learning algorithm, your input data is actually going to reflect your outcomes actually. If you are not giving a proper input data or training the algorithm in a best way, then outcome is also going to be a partially outcomes, I could say that actually speaking. That is what the input data is really important when come for data analytics as well as for the machine learning predictions. So I will train my data in such a way. First step, you have to data mining, learn about the data, input the data and apply the algorithm, train your data sets. So if I'm training for one specific task, so train it very well. Now, once it is done, your training is very well done and having a very good statistical, say for example, linear regression R square is equals to 0.98 or 0.97 or 0.87, something like that. So first when I apply, yeah, now my data is very good enough. Now what I will do, I will introduce a test data. That means unknown molecule sets. Unknown molecule sets. For example, maybe the second group of people want to test that molecules for toxicity profiling. So now I have, you can see the differentiations, right? I have introduced some different colors and the combinations I have introduced one in different colors, same pattern, but different colors outside and inside. You need to process it. The tool or an algorithm, what you train should have a potential enough to identify which pattern or fingerprint is making toxicity. So it will try to find out only that fingerprint talking toxicity. No worries, I will show you the demonstration. That time you will know much better. This is what I'm trying to explain to you all, because you all should develop a single any data like this. Then when you do the process, first thing is data mining. So take your data sets correctly and clean your data sets correctly. Try to add all the physiochemical parameters. We call it the descriptors or parameters or an whatever you call it actually. We call it the descriptors or physiochemical parameters, kindly like that. And when you apply the algorithm, it should be a potential enough to understand each and every properties of the structure and understand and more about it. It's just a training set. It's keep it in memory actually speaking. Now, once you process for the tested data, it should have accurately, it should predict the model and say this is a toxicity. This is what you call supervised learning, because you are supervising it and you're trying to tell the data about this process, then you're executing and testing the systems. This is called supervised machine learning algorithm. So we'll use this for the certain understand and then apply this. Next, we go for an unsupervised learning. So unsupervised learning, when I told this unsupervised learning will be using for clustering of the data or clustering of the structures or reducing, if I have a 2K during a supervised learning, I have ended up with an more than 2K physiochemical parameters, but all parameters are not going to significantly contribute for my systems. Rather, there are very few physiochemical parameters are very important for the toxicity profiling or activity profiling. So unsupervised learning will help you to reduce your or significantly help you to know what are significant parameters actually, or I could say this, it will let you know about what are the very important parameters for your process. So even when you given a raw input data like this, when you apply the clustering algorithm, for example, I am applying the clustering algorithm. It has an ability to cluster the inheritance patterns of the structures and try to group the molecules in certain spaces and let me know. Maybe including that, I am introducing A log P as one of the parameter, or acceptor as one of the parameter, or a topological descriptors as one of the parameter. There are certain groups of molecules will try to behave in the same pattern rather than the second group of people, second group of that one. So all these things will be clustered and let you know about where this particular patterns are clustered. So to know this process, we will apply the unsupervised learning technique. So finding the patterns or inheritance of the structures or clustering all this, you need to unsupervised. You don't have to supervise this technique. The tool itself will do all this process when you apply the algorithm and try to bring out the proper data sets. There we apply the unsupervised learning machine learning algorithms. Driving towards this is another level of the comparing to the supervised and unsupervised. This requires a little bit more training, I could say that, because this is called reinforcement learning actually. So reinforcement learning, it automatically determines actually. So it will let you know about ideal behavior of the structures. So I could say that it's to make a decisions in environment to maximize your cumulative reward. Or for example, what I could say that if I want to label the structure and I give the input data, it itself, the tool itself, or when I apply the things, environment, apply the algorithm, it rewards and understand the environment and then try to come out with the respective results. The core concept behind this is based on the environment which we are operating, actions which is trying to take it, or the reward it is trying to come upon, or the value functions. So this is the most important things about the reinforcement data. So this is what it's about, Anna. And this is actually a continuous learning process, and it will improve based on decision-making process. So this process keeps on repeating actually, reinforcement learning. So these are the three different categories. At present, this particular data are influenced, but data size is not small as mentioned. It is more than millions and billions actually speaking. Now, first we understand the problem statement. Now we understand that how you can take the different algorithms to process it. Now actually comes various in silico techniques. So our perspective and concept of lying to understand the toxicity profiling for the drug safety. So if you see the in silico toxicity analysis, you can see the expert systems. Expert systems are SAR, structural alerts, similarity, reach, RIGDOS, and these are the learning systems actually speaking. So learning systems are structure-based, QSAR, deep learning, machine learning, applicability domain. I will let you know as I keep working on. So these are all the in silico toxicological techniques. So today we more concept on this specific concepts actually speaking. These concepts we try to drive it more, because here you can get the, once you know how to develop the model, your work is much easier and you don't have to spend a lot of time actually speaking. This is where we are going to concentrate, learning systems rather than expert systems. So for example, so we are going to use learning systems. I have mentioned it. For example, the Biovia solutions. The Biovia solutions, how it is going to give a hand. Two ways you can do this process. One, as an open source, because some people don't have this kind of tools to perform it. Two ways you can perform it. One, you have to expertise yourself into certain scripting languages like R scripting, and including a bioconductor and RDKit and Jupyter Notebook. So these all are the core things will help you. Small about a Python codes actually speaking. So if you want to experience yourself to develop the model and build the model and execute the model, have this open source tools, RDKit, Jupyter Notebook, and small Python codes and respective libraries you have to install, and you have to perform the process. Other way around, like a commercially, now I don't have time, I have to execute the process. There are certain softwares or a commercial softwares are already existing, will help you to do the process much easier actually speaking. So if, for example, Discovery Studio is more than 3-decade software, will help you to know much better. So these are two different ways you can handle it. So now I will let you know about how to drive the process, and based on the Discovery Studio module, how you can predict the hepatotoxicity. And by giving the molecule systems, it will tell me whether it's hepatotoxic or non-hepatotoxic. And we drive slightly deeper into the toxicity profiling data where you can perform. So you don't have to create a models here. The models are built based on OECD principles and approved by FDA and NTP, national toxicity programs are like that. And where you can input your data and understand it. And on the top, you will come to know about using Discovery Studio Toxico4. Usually, we will try to do this process for an Pharmacophore. So here we call it a Toxico4. That means you try to understand the toxicological profiling of the molecule or a specific pharmacophore which is contributing for toxicity. We call it a Toxico4. And next is a target-based toxicity profiling and machine learning AI using a pipeline paradigm. These are the things, each and everything I will show you the demonstration so that you don't have to worry about it, how to do the process or where I will get the data or how to perform this analysis. First, whatever the tool is, we will build the model and we understand the model, we evaluate the model, then we test our model. Let us introduce a supervised learning technique here. So this is actually first I'll take you to target based, then I come for the other one. So target based things. Say for example, there is one drug. I'm taking up one drug. So second drug, this is a specific target for one specific primary target. And same thing can be a target for second one and for the third one. When it is using for second one, we call as a repurposing or repositioning. But the same drug is going to be on us giving you side effects. We call it off target. This is actually based on structure based or target based. How do you perform this? Say for example, when you try to work on the drug screening process, maybe take a breast cancer as an example. So ESR1 is my primary target, that is ESR alpha. Okay, this is a ESR alpha. You have to consider this. This is my first target. Same thing if it is a ER alpha. Estrogen receptor alpha, slightly the structure wise, it's going to share with the beta also. So I can use it for an secondary target of an estrogen receptor beta, and slightly it is going to bind with some other receptor. So based on the position of binding and how it is binding, where it is binding, all these things I'll be considering when I want for the repositioning or repurposing of my drugs. There are certain targets where I don't want my drug to go on bind. In the same physiochemical pathway or metabolic pathway, it should not go on bind. If it is binding, that will create a toxicity. Or if it is binding to my cytochrome P450 family of enzyme, that is going to affect my metabolism activity. So if you consider all these things, maybe hundreds of targets you can have, maybe 200 targets you can have, bring all the targets in one specific space. Bring all the targets in one specific space. 200 targets was one specific drug will help you to understand the rational drug designing and toxicity profiling. This is how you have to do the process. By doing this process, you understand or end up with the two solutions. One is you understand the repurposing of the drugs, and the other one you come to know about the off targets. So where and all my drug should not go on bind. You come to know about the off target. When you know the off target, try to change the functional group so that it should not go on bind for the long time. Or you can do molecular dynamic and simulation so that the drug should come out of within a 40 nanosecond. Very important, and most people use this concepts actually. When you do this analysis, try with your primary target over 500 nanoseconds, it should stay stable within your system. If it is off target, within 40 nanoseconds, the drug and molecule will separate. This is another way of finding out the target-based research actually. So it will let you know what is a conclusion you come to know. So there is a off target will present, and you come to know about way to reapply this drug, and this will help you to know the effective of drug and also toxicity nature, where and all to go and bind. And potentially you come to know about it, new network and multiple targets pathways. This phenomena can be briefed to not only for one single drugs, it can be used for multiple components and multiple drugs also. And this will let you know about various multiple targets and disease pathways and where should not go and bind. That's where the concept of polypharmacology will be applied. See the interconnections. We all know this concepts actually speaking. Only thing is we have to try to interconnect together to know more in depth of it. That is based on target-based toxicity identification. This is one of the method. Next one, it's going to be little mathematical modeling, not target-based approaches. So if I have a molecules, now we are having 2K molecules in your hand. First, we will try to understand the hepatotoxicity. So whether my drug is having hepatotoxicity or not. So what you will try to do, what are the current hepatotoxic drugs are currently in the market? Try to retrieve the data. So simple example, my concept to find out the hepatotoxicity of the drugs. Currently, what are the molecules up for there or databases there for the hepatotoxicity? Try to drive all the molecules from the database, not from one single source. Try to use a multiple sources and remove the duplicates. This is called cleaning of your data, and learn the physiochemical parameters and take the data, learn the data. That is what is most important. So now we will try to use a predicting AI modes. So there is a software in Discovery Studio. If you can give it, I will show you demo. So when you give a molecules inside the tools, it will let you know about whether having good absorption or bad absorption, solubility, good, low, or stable. All these things based on the categorical wise as well as in structural wise, it will predict and let you know about. This is a predicting mode. Now we'll pick only the hepatotoxicity. We will build the model and we test the model. So get into the ADMET hepatotoxicity. Say for example, there is a paper published by Chen and Dioxin. So in 2003, this is majorly to develop the model for a liver toxicity. See, along with it, you try to add a more number of data. Okay, if you want to develop of your own actually. So this is actually a model which has been taken from Cheng and Dioxin from 2003. So the molecules which he has worked on has been retrieved and they studied for this. And to generate the model, they are trying to use a classification. They use a classification of structure-active relationship and introducing a Bayesian model. That means what I want to know from this, you have to have very clear data. Hepatotoxicity. Hepatotoxicity. So when I give my data, when I give my input, it should say true or false. That means toxic or non-toxic. That's the reason we are introducing a Bayesian learning. Bayesian will let you know either this is a classification algorithm. It will tell you whether it is toxic or non-toxic, 0 or 1, binary. Okay, 0 or 1, toxic or non-toxic, true or false, like this. So this is what my output. I want output like this. So take your data like this. So first they will take the data, they will train the data along with I introduce a Bayesian categorization, right? So based on Bayesian categorization, I will get an RO score. Leave one out cross method is used. It's one of the validation technique with a recursive operating characteristics curves. When I train my data, I should get a good ROC curves. And with a good ROC curve, I will come to know about it, whether my system is good or not. In case if I'm not getting a good ROC curve, either you can add your data or you can remove your data or you can optimize on your physiochemical parameters. This is where you have to work on. So you can improve your quality of a model. You can introduce a system to learn a lot more about your molecule. Maybe that learning system is not enough to, not correlating properly with that. That might be a reason. Or the physiochemical parameters that you use cannot be matched with that or having some issues with that, because that parameter could have a positive effect or negative effect. Try to correlate with positive effects for this actually. So once you do this process, it will let you know about sensitivity, specificity, and concordance rates actually. So when you have a good data, it's good to want to test your molecules actually, unless this is where exact task is present. If you're not getting this proper validation, initial validation with training set data, so when you give a test set data, it will be failed. So try to work on the liver toxicity profiling, collect your data, understand your data, develop a statistical model, understand the statistical nature of that, then you test your models. This is how it processes like this. Data curation, very important process. So this is where the people will lack, actually speaking, especially the people from various background. So pre-processing of a data and data curation is a major process in a machine learning algorithm. Once you do the process, train and test a split, model building, and cross-validation with a hyperparameter optimization. So performance estimates, good or bad. If it is good, test data performance. If it is not, if it is a bad, go for the repetition of data. And include the cross-validation techniques like ROC curve is one of the cross-validation along with the leave one out cross method. Leave one molecule, test it. Leave one molecule, test it, like that. And performance of the model. This is where the actual machine learning process is present actually. So very important. Understand your data, split your data correctly. It is not, there is no mandatory that people will consider that whether I should have a more in a data set of training, more in test set, nothing like that actually speaking. It could be anywhere. Your training could have a more data, your test may have a less data, your test may have more data, your training may have less data, vice versa. Anyway, you can use that. So note, one has to aware of what should be given for estimating model performance. It's very important actually speaking. So you can introduce a various cross-validation technique to understand your performance of your systems. This is a simple example. So let me show you this demo, then we move on to the next one actually. That's where things will be fine. I will just show you the demonstration. So input data systems, I will take a systems of molecules. I will take it from samples. So this is how the Discovery Studio space will looks like. So let me create a new folder for you all. New folder, FDP. So there is a FDP I created. I'll set as default. So how many molecules are ended up I had? Here I'm taking 2K molecule, okay, approximately 3K molecules. So what my process I have to do? I have to do this makes you much easier, but behind it, people has to develop it. I want to develop a toxicity profiling. I will just select only the toxicity profiling, just for an example. So all molecules, given. First I developed, I have introduced, now I'm testing my systems, that's it. This is how we have to test it. So click OK. View results. Okay, I have not introduced one more parameter, that's why I'm not getting a graph. So here, you can see, I'm just concentrating only on this space actually. I'm free to move this. I'm moving like this. You can see true, 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 true. That means it is all toxic. Further, I will let you know because of which moiety is making toxic and all in a next demonstration. So true means it's toxic. So I can filter this. The toxicity prediction equals false. That means false means non-toxic. So I have took majorly 2.90 molecules, 903. Now if you look into it, I ended up only with 1037 molecules. So almost you can see that folds. One fold of molecules are being gone when I introduce hepatotoxicity predictions. This is very, very important. So this is all based on the ranges and properties. So applicability will be present. I'll tell you about applicability, you know more about it. This makes much easier. First, to develop the model, developing is a task. Testing is much easier actually. So this is what is about. So when you give it only the toxicity, I removed true or false because I introduced a Bayesian categorization, very important. That's why I'm getting only the two different options, true or false. True means toxic. I remove the toxic molecules. I have only one to the false molecule, that is a non-toxic, non-heterotoxic. So what is applicability domain? Majorly, it's one of the important parameter. So its objective to compare the newly predicted with the training set as was giving a confident about the new prediction. Applicable if it is within that specific region, within that specific limit, that is called applicability domain. So more you models trained on data sets that will try to create one chemical space actually. Say for example, it is creating certain space in this actually. If you have the molecules like this, okay. When I try to create like this. So all the molecules will try to have certain spaces like this. When the molecules outside this space, those are considered as hepatotoxic. Within this chemical space, it's considered as non-hepatotoxic. This is called applicable domain. Comparing newly predicted to the training set and the confident about the new prediction. How do you get confident? We'll consider that, okay, we are developing a model. Am I doing it right or not? That will be always a question racks your brain to understand more about it. That is called applicability domain. So aim to generalize a model, predict in a chemical space. It's an important thing to include OECD guidelines for the validation of QSCR models. So concept of applicable domain is in a novelty reduction as is for the confidence estimation. This is the most important one that you have to do it when you develop your models. Because first question, people will ask you, what is the difference between your model and the person who already developed the model? And what applicability you used for this, what chemical fingerprints you for this, what is not included in that. So these are things that you have to understand when you introduce applicability domains. This is applicability domain. So even good statistical model has limitations. Predictions will not be equally for all for good samples. More similar molecules in a training set, better the things. All these things has to be considered. This is called model applicability domain. So optimal prediction space should be more and trustworthy so that your molecular system, that system that you develop or the model you develop should be applicable for all the molecules. Globally, systems should be there. See the screening, so hepatotoxicity. So it is based on the one single data. So you can also develop based on the some other data, which has been highly predictable and usable. So true or false. So you can see the false. Wherever true, it's a true. So that means toxic. We have to take only the false. Next, we get into the next another technique. See, now you come to know these all are the non-hepatotoxic molecules. Still, I'm not going to close it from here. There are few more parameters I have to test because gender-specific models are more important for the toxicity profiling and for safety levels actually. So now what I'm going to do, I will try to introduce the learning system of not QSAR, rather I will introduce QSTR actually, quantitative toxicity relationship, structured toxicity relationship, QSTR. Rather than activity, we deal with the toxicity now. How do I do that process? The QSTR, it's a robust and cross-validation technique as like a QSTR technique. Rather than that, we will retrain the model, we understand the model. Usually what we will do, if you want to test the toxicity profiling, we take that chemical, we dissolve the chemical and ensure the concentration. We will do the biological transformation to understand toxicity. Here we don't have any wet labs, no experimental. I have only structure in my hand that told me these are the non-hepatotoxic. With that particular input system, I want to learn still more about toxicity profiling using a QSTR concepts. So that is where the mathematical transformation to toxicity. How do you do that? So query input structure, top cut assessment that we call as we do right now. So either it is positive or negative. So we will introduce a concept of similarity search as also machine learning algorithm into it. So similar molecules will have similar activity. Similarly, in the same way, the similar search when you do with the toxicity molecules that let you know because of which molecule or which molecule is making a toxicity. It's a very important concept actually. So I will show you the demonstration. So first we understand, then we go for demonstration. Okay. Here only we will introduce a concept called toxicophore actually. So what is a toxicophore? You have a seamless, can you see this? All are similar structures actually speaking. So here I have a bromine. Here I have fluorine. Here hydroxyl. Here I don't have just CH3 actually. So each and every position of the molecule is related to the structural property, or it may be for an activity or for the toxicity. So the part which is essential or responsible for toxicity, we call as a toxicophore. Very, very important one actually. So this is the most important concept that has to be dealt in the drug safety analysis. How do you do that? Say, let me show you this. I told you the same structure, right? This one I'm testing for an I will show you the demonstration. This is a input and output data to show you the differentiations between you people. So same structure. Imagine this is your structure you're going to take. I just changed this position alone, different halogens. And here I, instead of halogen, I place a hydroxyl OH group. Here I don't have any, just CH3. Let's see between these four molecules, what is the effect of the toxicity. See, this is for a female mouse. This is I took the female mouse. How do I develop? I'll show you in the software. This is for carcinogenicity testing for the mouse female. Based on the FDA data, the model was developed, applicability was done. Then I'm testing my system. This is how we'll be doing it. As doing so, I'm doing a similarity search also. This is what a similarity search will looks like. And you can see something called fingerprints. This we call as a fingerprint. That's what the importance of your 1D structure. Smiles formats are playing the major role in that. So this is showing the non-carcinogen. But same you can see here, this is showing the carcinogen. What is the difference? Instead of Br, here present OH. Because of this specific OH in this location, this molecule became toxic. When you try to treat the female mouse, female. So it became a toxic. Let me see the other two structures. So this is also became carcinogen. This is also became carcinogen. But the same structure, when I place a bromine, it is a non-carcinogen. Same structure, similarity, all almost similar. Only one single functional group, that is called toxicophore. So that's the concept of delivering about toxicophore. Single functional group replacement could have a great impact or more impact on your activity profiling as well as toxicity profiling. Since we are dealing with drug safety, you have to drive more on the toxicity profiling. So let me show you this one. So I will show you the demonstration. We'll understand a lot more about it. So let us have these structures. So I have, let me pick only the few molecules from here, just for demonstration purpose. So I'm taking only 4 molecules. Toxicity extensible. So visible molecule. We have a different models in Biovia. You have NTP, rodent, FDA, carcinogenicity, rat oral 50, all you can sensitization, oral irritancy, all you can have here. Let me take FDA. Click OK. Similarity search true. Detailed report, give it true. First time if you are running, give it true. Click run. So you can see the jobs is running. And it's preparing the report. It's writing the report. Same way, I will open the another client called Pipeline Pilot. This is a Pipeline Pilot client. So here also you can have that if you want to develop a new protocol or execute a new protocol, this is a Pipeline Pilot client. You can create a new protocol, input read your molecule systems, and then enumerator like-grade molecules. All you can do it here actually. So here already the tool program is developed. I'll show you how to do the process. Say for example, yes, I'll use ADMAT property of discovery, same discovery studio tools. I will use this from a different client called Pipeline Pilot. Toxicity extensible. So you can read your input molecules. So 20 different molecules I'm going to read. So I can read in all the models I can do with one model. If I don't want to do another model, I can disable and pass the data. You can disable or pass the data. This is how we'll work on machine learning algorithms, because we will develop it, but whatever it's required, we use it. Whatever it's not required, either we pass or hold the data. Let me run this molecule. See, passing, it's moving on, and you get a reports. Okay. So you can view results in Discovery Studio Visualizer. So this is one of the way, or you can do it directly into Discovery Studio also. Either way you can use it. That is why I'm showing that tool. If you want to change some data and you want to understand that one, Pipeline Pilot is the best tool, I could say that, where you can bring the changes. Here you try to use a user output. I mean, whatever data is inbuilt, you try to drive on the same. You cannot make any much changes, but there you will bring the changes there. So similarity report, detailed report. So see the fingerprints. So because of this specific fingerprint is a positive contribution for your molecule to be became a carcinogenicity. This is a negative contribution. That is a positive contribution. That's where the importance of these are called fingerprints. See, these are atomic fingerprints. If keep F, it's a functional fingerprints. Similarly, for this molecule to be a non-carcinogen, what are the positive things? And what are the negative contributions? So understand about the fingerprints is very, very important for the machine learning algorithms. Okay, let me take you to the presentation. So that's why I given input to molecular systems and then assessment. So assessment is most important. All the features in TopCat of Biovia is patent and has been enriched with the updated models. And if you want to do the same process in Pipeline Pilot, learn about it. So if you whatever you're doing in Pipeline Pilot, it's became a no-code process, actually speaking. But if you want to develop your own code, as I mentioned in the earlier of the presentations, try to learn certain scripting languages and statistical parameters and interface. You can use whatever the interface is best for you and launch your data and use it. Either way you can use it. So this is an example, whatever I have mentioned, so little bit depth into it. 2 types of toxicity classification models, good or bad. Regression models will let you know. Classification will tell you, yes, it is good, it is bad. So they are built based on Bayesian techniques. So how it will do? Input your systems, go sample by sample, good or bad, it will understand. We use this feature scoring. And finally, it will tell you about whether model is good or bad. How do you do that? For same model, you can use it for different types of studies also. Malgant versus non-malgants, or beginning spam versus non-spam. It's very simple Gmail filtering techniques, actually speaking. Active versus inactive molecules. So everything is based on your training set. It's a good or bad. Learn good molecules to support your predefined molecular descriptors. So your parameters is really important, plays a major role here. So if your parameters are not really good enough, or if you're not giving exact good descriptors, then the task is a little bit hard to have a quality of model. That chemical space that we are creating, right, that will become a little bit difficult. So learn the categories also. So which one you want to take it actually. So machine learning versus classification problem. So in classification, we count on area under the curve, recursive operating characteristics. So you have to check the performance of multi-class classification problems also. So use this is one of the evaluation method, that is a recursive operating curves, one of the evaluation method. It also, you can see in some papers, you can see area under the curve operating characteristics also. So leave one or crossword method. It's one of the validation of results we have introduced here. So any models that you're building, validate using leave one or crossword method is one of the good way of going in a cheminformatics technique. because many cheminformatics technique, we will see this leave or not toss out validation will be there, including a QSAR, 3D QSAR, whatever the models, pharmacophore models or 3D QSAR pharmacophore models, they will use a leave or not toss out method. So at what time, each and every time one sample will be left and model will be built, then will be excluded with the samples actually. So ROC curve is the most important parameter. Will let you know about what is the efficiency of the specific model, or we call it the area under the curve. The area under the curve must be above 0.7, above 0.8 is ideally good enough to move on for the further applications. So validation, see, this is an example of a mutagenicity. So when you have a getting area under the curve 0.8 means it's a good enough of 80% chance the model will be having a good enough to predict your systems. If you have 90%, that is good enough, 95% good enough. So more you improve your model, same way you have to test your model to ensure the efficiency is balanced correctly. So model building is literally a little bit hard task. I don't say it is a hard task. As you keep working on first model will be difficult. The next model will be ideally good enough to move on for the better validation process. So this is how we'll be doing with the pipeline palette. We read, take a screen read, SD reader or the molecular reader. We learn the system and we execute the systems. I will show you one of the example also. And we get in a specific or statistical parameters to understand more about the specific model. Then we perform this, for example, I showed you right now, right? So delimited text reader and then molecular text. So if you want, I will show you one of the live demo of that new protocol. I will take the SD reader from here. Copy, paste. Now I have the molecules. I can take any number of molecules from here. So now I want, so how the protocol is mentioning. So molecule to smiles, even I don't require that. I'll just take it out a component of toxicity. Topcat. So it's a whole developed systems actually speaking. And I'll try to view my models. Copy it, or how do you mention HTML molecular table viewer, or I will take only this model. I will try to take, okay, here at female NTP, HTML table viewer or report viewer. Anything you can see. I can move the pipes like this, anything. So let me read the input systems. I can read my source from here. Or SD reader, or I'll remove this also. SD reader. So input source, list of molecules. Let me take some molecules which I have already. Chemistry data. They don't want much molecules. Okay. Okay. I'll do one thing. I'll take only the 5 molecules. I will save it. These molecules, I will save it. Remove the attributes. I don't want these attributes. Click OK. Go to File, Save As. I put it in a public files. Biovia, PPS, public, SD format. That's it. Now I get access here. Shortcuts. See here. Sample drug molecules. Like this, you can select your molecules. Just run your systems. Okay, HTML viewer is not okay. That's okay. So can you see here? Input your systems and automatically says it's a toxic enrichment and whether it is prediction is true or false. It is toxic or non-toxic. True means toxic, false means non-toxic. Like that, If you want to screen for ADMET, if you feel that, so I want only NTP rodent carcinogenicity, this is how we'll be write the programs for this. We're using a pilot scripting. So in case if I want to include some other ADMET properties, say for example, with the same model, I want to calculate aqueous solubility. I will introduce aqueous solubility here. Delete HTML table viewer. So this molecules, I will read this specific parameter or hepatotoxicity. Yeah, this suits well for us. Hepatotoxicity, yes. Bring a model here. This is not refined. For those molecules, read only this. See, along with this, I am getting hepatotoxicity also. One more parameter added. False, false, false. Not toxic, non-hepatotoxic. For rat female, it is also not toxic. This is how we'll be develop the models. If you want specifically that, you can perform using a pipeline pilot. This is how that tool I was a sample example I showed you live demo also. So even you can apply this for understanding bad features and new features also. And classification, for example, I told you rodent carcinogenicity, aim mutagenicity. You can use this for skin sensitization, skin irritation, ocular irritancy. So various models you can develop, or you want to bring some external models also. Yes, that is also possible with a pipeline pilot. You can develop that scripting outside, convert into the component. Whatever I use is a components actually already inbuilt with the scriptings, and then the rules and everything will be there. Only when you give a molecule that will predict and let me know. So build like that, bring out like a component and easily perform the process. So I use a classification, and the regression is also is one of the technique. So where we use a continuous dependent properties. I mentioned regression analysis and supervised learning, right? So PLS method is one of the method. So what are the other ways you can use the regression? Optimizing a therapeutic ratios, prioritize my molecules, evaluate the intermediates, metabolites, screening the molecules, assessing for pharmaceuticals, commercial products, and you can get a better way, set dosage animal ranges like LD50, rat inhalation, toxicity, carcinogenicity potential, all you can able to develop. Anything you would like to create an output in terms of an quantifying data, we can use a regression models. Like I mentioned, TD50, LD50, EC50, minimal dosages like this. So same model, same molecule. I'm not going to change anything. Say for example, LD50. Okay. Let me take some, these are the different models are there, right? So let me take this regression models. Okay, rat or LD50. I want specifically only for this specific models. I can save file, save protocol as toxicity. Here I can save this protocol. This means customizing yourself with the toxicity profile, toxicity profile, something, or you can name it yourself. So that became toxicity profile. So I can close this too. I can open the protocol like this. So this is for customizing yourself with these protocols. Click and try and see here. Now I get rat oral LD50 also here. So that's how this molecule. So if you have a 2 lakhs of molecules within 20 to 30 minutes, you can drive the process and you can come to know about these parameters that will help you to prioritize the molecules. So set dosage, animal ranges, which dosage I have to start, all these things you have to work on the back and forth, and you can come to know about which is the best and which is the best good enough to move on based on toxicity. All you come to know about based on this analysis. So first, developing a model is a task. Rather, rest of the things are going to be only testing analysis. So this is a sample example. So of a different curcuminoids. So we did a analysis and what is the NTP, Ames mutagenicity, RAT PD50, RAT oral LD50, what is a units all given in the below, you can see here, and what is for fatted minnow and Daphnia and so on. So application, so you have seen about this one. So applications of AI/ML of TopCat, very simple, straightforward inputs, 2D structure. Only thing is in the 2D structure, when you are developing a model, understand the 1D format that will let you know about either the toxicological fingerprints or as well as a toxicophore analysis. This is the most important thing. So computation toxicological tools will rapidly help you to understand about your nature of your molecules. Also, you'll try to examine the toxicity relationship based on substructures, whatever you're providing, or the latest, what is the changes you have to introduce in the systems, and ranking them based on this for further experimental testing and also for defining the new molecules. So now that's a reason we have to develop a toxic-free molecular generations. Okay, another thing. Now you come to know about it. So my study is specifically based on only these molecules. I cannot change my systems. I have to go ahead with only that molecule. This is my parent molecule. I want to develop my model. Rather than developing a model, if I come to know this is making toxicity, I can remove, because I am an organic chemist or medicinal chemist of a pharmaceutical background. I know if what replacement of the group we could have a good yield in synthesis also, as well as I can remove the toxicity. This is your, if this is going to be your problem statement, then we will introduce a scaffold hopping technique. So in the previous structure I have showed you, right? Here, I showed you, right? This is a parent molecule. I just change a different halogen that has impact in your carcinogenicity. Same structure. Similarly, here I'm taking one parent molecule. This is my parent molecule. Because of this specific group, this is going to have a toxicity. So I am going to replace this specific groups now. What I will do, I will use a concept of replacement fragment. So I'll show you a demonstration also. I'll use a concept of replacement fragment, remove the specific fragment, try to introduce as much as different fragment from the libraries. So libraries are inbuilt, you don't have to worry about it. User generated library also can be given here actually. So try to generate the different fragments and let me know whether it is having toxic or non-toxic. This is program also you can automate in pipeline pilot. So if it is fine toxicity, remove that parameter and exit. This is how the pharmaceutical companies will execute. Because once you give an input, they want only the output. They don't want to make any human interventions to do this process. Rather, they will automate the process. That's where the pipeline pilot majorly plays a role actually. So input your molecule. Now we will try to replace it. I will show you the demonstration of this. Okay, I will close the systems. Rather, this is a scaffold. So now when I come to know this is a place toxicity is present, I want to replace it. So replace fragment. So I will take an input molecules. Okay. So this is a functional group which is making. So for example, you can select from here also. If you are very well known about the process, then you don't have to worry about it. Select up to this. Let me select for you once again. Okay, I selected the group. This group I want to replace, for example, input ligand, fragment to replace, create group from selection, click around. That's it. So now the toxicity fragment will be removed. New fragments will be generated. For that, you test the toxicity profiling and take which is the best and move on for the further process. This is called replacing your fragment process using a scaffold hopping. Whenever you come to know in your structure is having toxicity, remove, replace, test, and go further for your analysis. It's taking some time. Okay, it will finish it off actually. Once you finish, You get results like this. So number of fragments used. So how many fragments are generated with the same fragment, and the output ligands try to do this process. So we are come to an end actually. So all this analysis, either machine learning or target prediction or toxic4 or scaffold hoping, whatever is it, all are directly contributing for the drug safety actually. That is the most important one. So any drugs which you are designing, one side you try to optimize for the activity, on the other hand keep an eye on the toxicity profiling also, because it's most important. So to do that process, one way you are trying to use a graphical interface software, on the other hand try to introduce a machine learning algorithms. Our AI technique will be feasibly will try to identify the which is a good molecules to have a toxic free environment, so that, because anyhow the drug designing and development is not at all easy process, it will consume more time, but making a cost effectiveness is a most important one. So optimization level, it's an iteration process to identify the promising molecules is one of the major tasks. This is also, you have to improve the biochemical potency, selectivity, bioavailability, kinetics, and reduce the toxicity profiling. It's the most important parameter. And efficiently practical computational tools will give you more hand along with your AI ML. And either you can do structure based or a component based or fingerprint based. It's up to the you people actually, which one is feasible for you. And you scaffold hoping, it's keep it as in another step where if you have to work on only that molecule, replace the toxicity and then work on the new molecules to ensure that you have a toxic free molecules for the compounds for public health that will have a rich ecosystems, enhance your research and prevent drug failure for the better public health. So hope I have delivered a few inputs to you all, added to your colorful to your wings. So let me know if you have any queries. Thank you so much for patiently listening to this demonstration as well as the talk. Thank you so much. If you have any queries, we will discuss.
Thank you, madam. Thank you very much. Actually, what we are intended for the FDP, you are given more than that, madam. With including demonstration. Thank you very much, madam. Dear participant, if you have any questions, queries, please post here in the chat box, or you can ask in the unmute yourself and you can ask.
So if you have any queries pertaining towards this or anything you could like to add upon to your works, also you can let me know.
Madam.
Yeah, somebody is. Yes, sir, please.
Yes, sir. Somebody raised their hand.
Yeah, Mr. Karthik, sir, you have raised your hand. You have any queries, you can unmute yourself and you can ask us.
Yeah, ma'am. That was a very detailed and self-explanatory stuff. Knowledge that very few are privileged of. Thank you for the wonderful presentation. But I have few, this is just out of curiosity. It's not doubts or not doubting the validity of the science behind it. But when you are performing all this kind of molecular dynamics in these simulations, and we are depending on structures primarily from crystals. So, but we practically know that most of the crystals are not, full crystal structures is not possible. Most of them are truncated proteins. So through practical experience, I have seen that these truncated regions are IDRs, intrinsically disordered regions. So the machine learning, will it help us in predicting these ideas? Even though we have AlphaFold and Rosetta, but how far we are in determining these unknown idea structures through AI and ML?
So thank you so much, sir. Actually, this is highly valuable and then valid questions, actually speaking, because when you work on a drug designing and development, we will try to work on the single receptor. Say, for example, estrogen receptor alpha and beta, but inside our systems, it is not like that. It is a combination of proteins will be present and try to do all the biological activity in the back end. In some proteins, you may not see the full structures will be present. That's where the plays of an alpha fold and D fold are playing a major role. On the top of it, certain things I would like to add upon. Without knowing anything and then finding out where it is present, these kind of structural biological tools or crystallography tools will give some insight about it. That's where we are actually playing the certain roles. Instead of the maybe my drug designing and development going to be a success or failure, that is the other end, but going in a blind way of finding out the results, these kind of tools will let you know whether it will happen or not happen, first thing. And lots of machine learning algorithms that currently people, how we work on the small molecules, there are certain group of people is majorly working on this kind of a biological also. That means even one of the protocol in Discovery Studio also, when you try to take X-ray crystallographical structure, it is not fulfilled. Maybe there is an atom missing. There may be a loop is missing. There might be not a proper side chain alignment. So when we try to introduce these tools with the prepared protein, it's one of the tool, will drive with the help of force field parameters along with the pH. These two parameters are part of it. When you do the process, instead of reaching an 50% probability of results, this will try to improve the probability of results to 90%. So that loop will be built, side chain will be built, loop will be optimized, and ensuring there is no structural hindrance or steric hindrance, and try to deliver you the best structure. With that, you will start with the process. Is it
Well
fine, sir?
said. Yes, ma'am, I totally agree with you because rather than having no idea about it, we are trying to improvising it, and that's a great job. But from industrial point of view, uh, I'm just wondering how far we have put in our effort. Say there are multiple multimeric proteins which
Yes,
might
sir.
act as a receptor. So
Mhm.
when we are taking, a truncated protein where the loops or a bigger regions are missing, and we simply perform a protein-protein interaction and consider that to be a receptor, will this give us a proper active site, or is there any ways to, come around these missing parts?
Thank you once again, sir. Any structure, say for example, when the translation process is going on and you get one domain of the protein, domain structure, when something is missing in the domain, we can model it using a homology modeling technique, or you can rectify the problems in your structure by introducing a structured biological tools and ensuring it as equal like a in vivo. First one. Only with the completed structures. That's a reason knowing your target is important. Target validation is very, very important process. From which region to which region? Say, for example, binding at 220 to 320 region is very crucial for the biological activity rather than the other location, maybe a C terminal, N terminal. If you know this process before you're starting, that only will help you to get the best results rather than believing on the tool wherever it binds. I will go with the process. That is actually not a right way of doing the drug designing and development at all. So you have to understand the biological process behind it. Accordingly, you have to drive your results. Then that kind of results only you can, it will help you in experimental technique that will improve your certainty and the productivity of results also. You must correct all the stuffs before you get into your, even when I show this kind of tools, pre-processing of your targets are really important. when the people do this kind of process, they simply take the structures and they do it. That is not actual drug designing or identifying a toxicity at all. Clean, understand it, then drive it correctly. That only will help you. Even when you do this process, there is always a 5% uncertainty will be present. To overcome that only, we are trying to do all the precision techniques or precision analysis, ensuring to achieve the process. You have to do this process, ensure that structure is present, that domain is present, model it or introduce using a machine learning algorithm, develop it, then you execute it.
Thank you so much, ma'am, for very precise and relevant point. And thanks for the patience.
Yeah, sir. My pleasure, sir. Thank you so much, sir.
Divya madam, one small clarification.
Yes, sir.
We are doing now molecular dynamic studies. In that, some people are doing for 50 nanoseconds, some are doing 100 nanoseconds. So, can you give clarification which what is the time if you are doing good to get good results?
Thank you so much, Dr. Sham, sir. When we come to the molecular dynamic and simulation, I think past 5 years, I'm seeing that people will think that only dynamic results is fine enough to predict your outcome. But actually speaking, that is not a right answer. And doing 100 nanosecond is going to give you the perfect results. Actually, that is also not a right answer. When you perform this, why we are performing 100 nanoseconds, why you performing 500 nanoseconds or 1 milliseconds, it entirely depends on the nature of the protein. For one protein to get fold or one atom to get fold or unfold, there are certain period of time it's required. Maybe if you look into the systems, if a one loop to turn from one location another location, so these many times are really required to do the process. Based on that analysis only, people framed that 100 nanoseconds is a minimal time to understand the process. And depends on that time. Nowadays, you can see that how you do experimental techniques like triplicates. Here also, you have to do triplicates. Why triplicates? At one point of potential surface only, your dynamics will start running. Second time, it is not necessary. It should start with the same position or same time at the place. If you do the second time, it might be pick at a different location of potential surface area and start running it. Maybe the third time, it might be taken a different potential surface area. All this in potential surface area has to be calculated in terms of triplicate manner. Then only you will drive it, you will come to know about it. There is no specific time limits like 100. As you keep running for more nanosecond, your results, not a results, you come to know about more on the protein folding and unfolding process. How it is binding, where it is binding, or they really strong enough to move on over a period of time or not. All these questions will come across when you run for longer time. At present, people are referring to 100 nanosecond, but that alone is not enough to draw the results. And only the molecular dynamics is not only the tool to give you the perfect results. That's what I was trying to mention here. Along with it, you try to add upon lots of other parameters and ensure to correlate with your results and produce your results. That's how you have to go on, sir. There is no specific seconds to finish your results. If your system, as I mentioned during a target things, at 40 nanoseconds, if your system is disintegrated, that is not at all a stable system. You run for another 100 nanosecond, check that stability is attained or not. That's where you'll see the difference between initial structure to next structure using RMSD. That RMSD profile alone is not enough. People are mentioning that only RMS radius of variation and RMSF is fine. That is not the actual way of doing it. It is telling you as a function of time what is the effect of its movement. But you should analyze in terms of a binding site interactions. When OH to NH is bonded together, what is the effect of the bond length? Whether it is stable or non-stable. That's how the molecular dynamic results should be there. And nowadays, you can see that papers are getting rejected because people are pertaining to show only these results that is not actually going to help it out. Try to add more parameters. Let's keep it as a supporting parameter. That's what.
Thank you, madam. Thank you very much. And one Kiran has
Yes.
raised his hand. Karthik. Please,
Karthik,
Karthik.
sir?
Yes, ma'am. So I think will replica exchange molecular dynamics will be helpful in this place where you have a lot of replicas move from one potential energy into the other, to answer previous professor's question.
Yes, sir. Yes. Most important potential surface is most important for that. That's the reason we are doing triplicates. But when you take a triplicate, take an average of that, then plot only one graph.
Okay. So could we bypass the triplicates by performing a replica exchange?
No, sir. Replica is important actually. When you do the three replicas, you come to know
Hmm.
what is the effect of the systems.
True.
Because that's the reason. If I told that may pick at F1 position, that may pick up at an F5 position. We are not known about it actually. So molecular dynamics and simulations is a stochastic effect actually. We don't know where it will pick, how it will pick. That's the reason potential boost surface area is really important.
Of course, yeah, we cannot depend on non-stochastic as Monte Carlo also in
Yes,
this
sir.
regard.
That's right. That's right. Actually, I agree with you.
Okay. Thank you. Thank you.
Thank you, sir.
Madam, now we have told that toxicity prediction tools.
Mhm.
Is it available in Discovery Studio Visualizer? Madam, is in commercial only.
Sir, all these things, see, whatever I have mentioned, that's the reason. These all are commercial tools, actually. There are few tools in case that if you want to develop the model, see, machine learning, developing a model is one of the prime area now people are working across the world. You don't need to have a commercial software for that. Only thing is you should have I could say basic knowledge of developing a mathematical model. All are available in open source. You can take some courses out of it and you can develop the model and you can test a model, not only one model, different kind of machine learning model. Or you want to based on the structural base you want it, that you have to work on the commercial perspectives that only give you the, somehow you can reproduce your results in experimental technique. That's how it works. It is not there, sir, actually.
Thank you, madam. Thank you. And on behalf of all researchers and who are working on computational studies, madam, and we are very thankful to you and Altem Studies, Discovery Studio. So many people are using visualizer, that is Discovery Studio only. So we are very,
Yes, sir, I
it
understand.
is very helpful tool to all, madam. And finding that active site prediction. So it is a very, very helpful tool, and everyone is using the Discovery Studio.
Yeah, sir.
And it's our lucky, madam. We are here. Sir, please ask the question.
No, the Divya madam.
Yes,
This
sir.
is Dr. Manjappa, and thanks for giving madam a lot of insights about AI, how we can predict and all. But what I want to ask is there any possibilities of or any case studies where software has showed non-toxic or safe, and when it enter the clinical phase, it's showing some kind of toxicities? Is there any such kind of cases? Yeah, instances which happened.
Sir, all these things, we are discussing in academic perspectives, and when it comes to the commercial perspectives, so same kinds of tools has been used worldwide, actually speaking. I could mention that AstraZeneca or many people across the world is using this kind of tools. And certainly the input models, that's what I mentioned earlier also, the input that you are using for this tool or the data you are using for the tool must be very clear. Based, if there is a change in your input data, there will be a reflection in output also. As of now, whatever we have come across from the various industries and other people, 95% there is a good probability to move on. There is always a 5% uncertainty is there. So 95% you can use it. There is no issues, nothing has happened because data is taken from an experimental technique and build the model. There is no doubt in that. You can go ahead, or you want to test your systems with the already existing toxicity profiling also, that also the tool has passed in that. There is no doubt in that. 5%, there is always uncertainty, sir. to be very honestly to put forward.
Thank you, ma'am. Thank you.
Thank you, Dr. Manjappa, sir.
Dear participant, if you have any doubts, please ask.
Ma'am, if molecules are very potent, but they may be having some toxicity issues, and is there any other possible methods we can minimize the toxicities other than changing the functional groups or something?
Other than changing a functional groups, so you can go for the regression based analysis and using a regression based analysis. That analysis will let you know the concentration there. From that concentration, either you can reduce the concentration to the minimum level, or you can dilute the concentration to the different level that you have to do experimentally, and through that you can enhance your process. So here the place of concentrations will play a major role here. Through that you can achieve that, or you can add some other another component to that as an additives or something. Through that you can reduce the toxicity. These are the two ways for the existing things actually.
Okay. Once again, ma'am, thank you so much for accepting our invitation, and I express my heartfelt thanks on behalf of management for accepting and sharing your valuable time.
Yeah, thank you so much, Dr. Manjappa sir, and then Karthik sir, and all the participants over here. And I believe that this kind of platform will really give us even insight for us to deliver what we are learning in the industries actually. That will help you to drive into more into academics, because from the academics only we are born and we have been come across to this particular level. So when you do this analysis and correct these things during in the college level, then outcome will be much better and best in future.
Yeah.
So thank you once again, sir, for inviting me in this wonderful virtual platform and to meet everyone across the country. And I thank the management and ask Mr. Dr. Sham, sir, for inviting me for the wonderful place where I can deliver my thoughts and then propagate the things over in this platform, sir. Thank you once again, sir.
Thank you. Thank you so much.
Thank you, madam.
Thank you.
Thank you.
Thank you. Thank you.
Dear
Sham sir,
participants,
can I leave the session, sir? Sham sir? Yeah, thank you, sir. Thank you so much.
okay, madam, you can leave.
Thank you, sir.
Thank you, madam. Thank you very much.
Thank you, sir.
Dear participants, regarding the feedback link, we are providing at the end of the FDP. So for your convenience, you can fill that feedback blank at one time and you can submit it at the end of the FDP, means on the last day of FDP.
Dear participants, thank you for your patience. With this, we conclude today's FDP session. Tomorrow, we'll meet at the same time, sharp 10:30 a.m. Thank you all.